I'd like to thank everyone for joining today's webinar. It is part two of a series entitled Real-Time Cellular Target Engagement. In today's webinar, what I'll be discussing is the CellarisBio microtag system applied to cell target engagement. I'll provide an overview of data generated with real-time instruments using the microtag technology and talk about different ways to analyze the data. And I'll present a couple of case studies where we worked with some challenging drug targets. So the CellarisBio microtag system is based on enzyme complementation of a split RNase enzyme. This RNase enzyme is composed of two subunits, a small 15 amino acid subunit that we call the microtag, and a larger subunit that we call the microtag binding protein. When these two subunits associate, they form an active RNase enzyme. For this system, we employ a FRET RNA substrate that is depicted in panel B that has a donor and a quencher fluorophore that are close together on a small or short stretch of RNA. This RNA is cleaved by the active RNase, then separates the two fluorophores, resulting in fluorescent signal being generated. We've applied the microtag system for cell target engagement, and what is involved is a thermal shift assay. So for thermal shift assay, a heat challenge is applied to the cells expressing the protein target. And what happens when you apply the heat is the protein target starts to unwind, denature, and form insoluble aggregates. And when it forms insoluble aggregates, this 15 amino acid tag, if it's cloned to the carboxy or N-terminus, will get buried in those insoluble aggregates and no longer be amenable for enzyme complementation with the large subunit. So the microtag system is specifically designed to quantify the amount of protein in cells. And what I want to depict with this slide is that the fluorescent signal generated from the assay is directly proportional to the amount of protein expressed within those cells. For cell target engagement, it involves a thermal challenge or heat being applied to the cells expressing the target. So in this case, where we have a microtag target expressed in cells, looking at increasing the temperature, so starting at about 38 degrees, increasing the temperature down to about 62 degrees. And what you can see is as you increase the temperature, you get less and less fluorescence being generated, up until about 60 degrees where very little fluorescence is being generated. So what's happening is as you apply heat to these cells, this protein target is starting to unwind, denature, and again form these insoluble aggregates that buries that microtag. And so for conventional cell target engagement, what is involved is identifying the midpoint between the low temperature and the high temperature and identify a temperature of melting. And in this case, it would be about this one right here, about 48 degrees. So then for conventional cell target engagement, you would take the cells expressing the target, combine it with an inhibitor or a test compound, and treat at that specific temperature, 48 degrees. And in the presence of a binding event, so let's say that ligand binds the target, what you would expect to see is that target will then be protected from denaturation in the heat applied at 48 degrees. What I want to really demonstrate in this slide is this target starts to unfold and unwind at about 39 degrees, or let's say 40 degrees, down to 60 degrees. So really what you want to be able to do is assess or interrogate ligand binding with this particular target from 40 degrees down to about 60 degrees rather than choosing one specific temperature. And so we've designed the microtag system to specifically work with real-time instruments. And the power of using real-time instruments is that you can utilize the heating and cooling programmability of these instruments to now interrogate binding events in a temperature series that we call a step gradient. And so this slide depicts a workflow of the microtag cell target engagement system where you would engineer cells to express a microtag target. So you would clonally add the 15 amino acid tag to either the N or C terminus of your target, express it in cells, grow it for 24 hours, and then transfer these cells to a PCR plate, add inhibitor or test compound along with the detection reagents, and run a step programming. And in this case, it looks like 40 degrees up to about 70 degrees. And this is what the data would look like. So for the step programming, introducing a temperature series to monitor or interrogate ligand binding, this is what is involved. So basically combining all the reagents with the cells with the test compound, ramping the temperature to the first temperature point and holding for a defined period of time, and then lowering the temperature back to 25 degrees to allow for detection of the amount of protein that's remaining after that heating event. Then raising to the next temperature in the series, holding at that temperature, lowering back down to 25 degrees, and detecting the amount of protein to quantify the amount of protein remaining after that temperature hold. And then heating again to the next temperature in the series, holding there at that temperature, go back down to 25 degrees, and detect the amount of protein after that temperature heating event. Now, you can introduce as many heating steps as you like with this programming. Also, you have the ability to change the amount of time that you hold at that particular temperature. And you can also modify the amount of time needed to detect your signal based upon the kind of target that you're working with, where some targets that are poor expressors, you may need to allow longer period of time to detect a protein. So this is a very powerful system that allows you now to test or interrogate drug binding to a target in a temperature series. So the key advantages of the microtag step gradient system over conventional cell target engagement systems are: this is a mix and detect simple procedure that uses real-time instruments for detection of a fluorescence signal. Now, the beauty of using the real-time instruments is that it allows you to program heating and cooling steps that allows you now to be able to test these different temperatures with ligand binding. So there's no more need to identify a specific one temperature of melting for your target protein and test at that one temperature. A single well will be programmed to go through multiple heat cycles followed by protein detection. And the amount of fluorescence that's detected really is an accumulation of all the binding events for all the temperatures. This is a streamlined workflow, fewer procedural steps, which saves time and money. And this is what the raw data would look like in the left-hand side of this slide on the left panel. So in this case, we're testing a compound with this target and comparing it with the DMSO control vehicle. And we're interrogating from 40 degrees to 70 degrees heating. And for this particular detection step, it's 10 cycles, and each cycle is 30 seconds. So this is 5 minutes from start at 40 degrees to 45 degrees. Now, what you can see from this particular raw data is that there's a separation between the DMSO control and the test compound at about 55 degrees. So after 55 degrees or at higher temperatures, the DMSO control, it starts to plateau off. So what is happening here is the target protein is denaturing and forming insoluble aggregates. But in the presence of a test compound, which is the black line, you start to see an increase, a continual increase in signal or fluorescent signal, which is due to the stabilization of this microtagged target by this test compound. Now, there's a couple of different ways to now look at this data or analyze this data. One is to look at the area between the two curves, between the DMSO and the test compound, as a measure of the amount of stabilization of this target protein with this test compound. Or you can look specifically at each individual temperature heating event. So at 55 degrees temperatures, you can look at the slope of signal generation for the DMSO control and compare it with the test compound to identify differences between the two. And this is the right-hand side of this slide, where you look at where you can identify or determine the slopes for each one of these temperature points and compare DMSO with the test compound. And what I want to point out here is that for this particular target, the two temperatures that are most critical are 55 degrees and 60 degrees, where in the presence of the test compound, the slopes are nearly double that of the DMSO control. So with this data, that was a hit validation type of data, but you can also introduce a dose series to identify a dose response for a particular compound, allowing you to determine an EC50 of binding. So in this case, we were looking at, or what I'm presenting here is PLK1, polo-like kinase 1. It's a microtag target that we tested a compound for binding to this target using the step gradient. And what you're looking at is the raw data output from the real-time instrument. And this is typically what it will look like. And in this case, it was a heat event for 3 minutes, followed by a detection for 10 minutes, starting from 40 degrees up to about 70 degrees. So now you can transfer this data from the real-time instrument to GraphPad Prism, and this is what the data would look like with technical replicates for error bars. And there are two different ways to analyze this data. One, as mentioned previously, would be the area under the curve for each particular dose and comparing it with the DMSO. Is a red line, which actually is under this purple line. Or the other way would be to look at the slope of signal generation after the temperature at which there is separation of the signal. In this case, it would start at 45 and continue. It looks like 50 degrees. There's good separation between the doses. So for area under the curve analysis, you would use GraphPad Prism. So this can be done with that program. Determine the area under the curve for DMSO, determine the area under the curve for each one of the doses, and then subtract out the DMSO AUC. And what you would be left with is this light blue shaded area. So this is a measure of or a quantification of the degree of stabilization of this particular dose for this microtag target. So it's an accumulation of all the signal for all these temperatures being tested. Now you can do that for each particular dose, determine the area under the curve, and plot that on a y-axis. And on the x-axis, plot the dose of inhibitor being tested on a log scale, and then fit a sigmoidal dose response curve. And in this case, the curve fitting results in an EC50 of binding of about 1 micromolar determined for this particular compound with this particular microtag target. The other way to analyze the data would be to look at the temperatures that are most impactful for the binding events. So in this case, you notice at the higher temperatures, it's a rather flat line for both DMSO and the higher doses. But at 45 and 50 degrees, there's significant separation of these curves. And so what we can do is use linear regression analysis to identify the slope from 45 to 55 degrees, and then plot that on the y-axis, and then versus the concentration of the test ligand or of the inhibitor being tested. And in this case, on the right-hand side, we're looking at cycles 21 to 40, which is actually the 45 degrees Celsius heating temperature. And we end up with an EC50 of binding of about 846 nanomolar for this particular compound with PLK1, looking at the slope or fluorescence generated per signal. So the data I presented was for a well-behaving kinase that generates very good data in the microtag system. But as anyone knows who works in cell target engagement, every particular target you work with behaves differently. And there are some that are more challenging than others. And some of the more challenging ones are multipass transmembrane proteins, such as GPCRs. And others that are challenging are intrinsically disordered proteins, such as transcription factors. So we've worked with a number of GPCRs, but one of the GPCRs that we had trouble with was GPR 75. And for this particular construct, we generated a microtag version of this construct that has the microtag at the carboxy terminus. So this is the intracellular part of this protein. Expressed this into HEC293 cells. And then we onboarded a number of antagonists. So these are well-characterized, well-described, commercially available antagonists to test for binding. Now, when we tested for binding with this construct in our step gradient, we could not detect any binding event. So these false negatives, I want to comment that they are common for thermal shift assays because these thermal shift assays, they all rely on the fact that once you get a binding event, that binding event needs to be potent enough or strong enough to impart a conformational change that can be detected, that can impart a conformational change that can modify the stability of this protein in a heat challenge. So in this particular case, these antagonists, although they were binding to the target, they were not inducing a large enough conformational change that can be detected upon a heat challenge. So how did we deal with this particular construct? Well, what we wanted to do is first of all, determine whether GPR 75 works in our microtag assay system. And the way we do this is we have in-house, we have a pool of 10 promiscuous compounds that we test during our assay development phase for our targets. And this is a step gradient that was run with GPR 75 in the presence of increasing concentrations of promiscuous compounds. And so what you can see is that at about 45 and 50 degrees Celsius, there's separation or stabilization of the signal in the presence of 1.25 micromolars of the promiscuous compounds, and still further increase in the presence of 5 micromolar compounds. So what this data tells us is GPR 75 microtag construct behaves very well in this assay and can be used to screen compounds for binding molecules for this particular construct. So another challenging class of families are transcription factors or intrinsically disordered proteins. And you can imagine how difficult it would be to look at a thermal challenge to a protein that exists as that is intrinsically disordered. So imagine placing a microtag on the carboxy and terminal part of this protein, applying a heat challenge, and then trying to look at ligand binding events that would stabilize this protein. That would be pretty challenging. One of the proteins that we worked with, one of the transcription factors we worked with is c-Myc. So this is an intrinsically disordered protein. We generated our carboxy terminal tag of Myc, so having the microtag on the C terminus, expressed this in HEC293 cells, and we have an anti-microtag antibody to demonstrate the expression of the tagged Myc construct. And then we applied a heat challenge, so increasing temperatures to see what happens to this construct. And so what you can see is with increasing temperatures, we start to get a decrease in fluorescence, but it starts to stall at high temperatures. What I want you to notice is it doesn't go back down to zero. You still get some fluorescence from at high temperatures. However, there is enough of a window here for us to use this for looking at target binding events or looking at cell target engagement events. You can take each one of these, the fluorescence at 60, at cycle 60, plot that against temperature, and it identifies a temperature of aggregation for Myc at about 47 degrees Celsius. So this is the conventional method, looking at identifying a temperature of melting for the protein. But what we wanted to do was to look at Myc in our step gradient. We onboarded a commercially available Myc binding molecule called MycI361 and ran a step gradient. And here, focus on the bottom right. This is 156 nanomolar of MycI361 So the DMSO is the black line, MICI 361 is the red line. So the overlap showing there is no difference between DMSO or the presence of this compound. As you start to increase the amount of this compound, 300 nanomolar, 625 nanomolar, and up to 1.25 micromolar. So now you start to get a separation of the curves. So that with DMSO, you still get an increase in fluorescence generated, but in the presence of MICI 361, you start to see a decrease in the amount of signal, suggesting that this is acting as a destabilizer for MIC. With further increase in temperature, with increase in the amount of the MICI 361 to 2.55 micromolar, you don't get further decrease in signal, suggesting that you've saturated the binding with MIC. So this data demonstrates that the step gradient with MIC, an intrinsically disordered protein, uh, uh, can be used to assess binding of a ligand to this particular target. So in summary, the microtag system that CellarisBio has developed is designed to work with the real-time instruments, and this has the advantage over conventional methods of cell target engagement in that you no longer have to identify the temperature of melting and interrogate drug binding to this target at that particular temperature, but instead the step gradient allows the testing of a temperature series with a single dose in one well. The analysis of the raw data from the real-time instruments can be done by looking at the slope of signal generation after every temperature being tested, or you can look at area under the curve, which is an accumulation of all the binding events over all of those temperatures that are tested. The microtag system has been designed to work well with challenging targets such as GPCRs and intrinsically disordered proteins such as transcription factors. I want to thank everyone for joining today, and I hope this webinar was informative. And stay tuned for further webinars from CellarisBio.